Bem-vindos ao nosso espaço, um mergulho profundo nos assuntos mais interessantes de esportes, ciências e temas gerais do momento. Aqui você vai encontrar conteúdo de qualidade, com análises empolgantes e descobertas fascinantes que vão expandir seus horizontes. Nossa missão é trazer informação relevante de forma dinâmica e descontraída. Queremos que você faça parte dessa jornada incrível de conhecimento e diversão. Se gostar do nosso conteúdo, não esqueça de deixar seu like e se inscrever no canal para não perder nenhuma novidade. Ative o sininho também. Aproveite!
If you love science, don't forget to like and subscribe. When we first mapped the human genome at the turn of the millennium, we expected to find a concise, elegant manual for building a human being. Instead, we found a chaotic library where nearly 98% of the books seemed to be filled with gibberish. For decades, this massive portion of our genetic blueprint was dismissively labeled as junk DNA. Biologists assumed it was merely evolutionary detritus, the scarred remnants of ancient viruses and failed genetic experiments that had hitched a ride across millions of years. But nature rarely wastes energy on such a massive scale. As we dive deeper into the realm of bioinformatics and information theory, a startling new hypothesis has emerged. Our genome isn't messy, it's redundant by design. This brings us to the fascinating intersection of genetics and computer science known as the Hamming genome. To understand this, we have to travel back to 1947 at Bell Labs. A mathematician named Richard Hamming was frustrated by the unreliability of early computers. In those days, machines read punch cards, and if a single bit was misread, the entire calculation would crash. Hamming realized that if you could build a mathematical structure of extra data around the core message, the computer could detect and even repair its own errors. He invented what we now call Hamming codes. These are error correction systems that add specific parity bits to a data stream. These bits don't carry the message itself, but they provide a mathematical checksum that allows the receiver to reconstruct the original information even if parts of it are corrupted. Now, consider the cell. Every time a cell divides, it must copy 3 billion base pairs of DNA. This process is remarkably accurate, but it isn't perfect. Environmental factors like ultraviolet radiation, chemical toxins, and even the heat of our own bodies are constantly snapping DNA strands and causing mutations. If our genome were a lean, 100% coding document, every single mutation would be catastrophic. It would be like trying to run a computer program where every single bit is critical. One mistake and the system fails. Life, however, has persisted for billions of years despite a constant rain of genetic damage. The reason lies in the 98% we used to call junk. This non-coding DNA acts as a sophisticated biological error correction code. In information theory, this is known as channel coding. By surrounding our vital protein-coding genes with vast stretches of non-coding sequences, evolution has created a mathematical buffer. Many of these non-coding regions, such as introns and repetitive elements, appear to follow statistical patterns that mirror the error correction protocols we use in satellite communications. These sequences act as a biological noise floor that absorbs the impact of mutations, preventing them from hitting the essential instructions. Furthermore, research suggests that the physical structure of non-coding DNA helps manage the signal-to-noise ratio of the genome. Some non-coding segments act as spacers, ensuring that genes are positioned in a way that minimizes the likelihood of physical damage during the tight coiling of chromosomes. Others act as regulatory check bits, providing the context necessary for the cell to recognize which parts of the code are authentic and which have been corrupted by viral insertions or replication slips. We are beginning to see that the junk isn't just taking up space. It is providing the architectural integrity required to store complex information over geological timescales. This shift in perspective transforms how we view evolution itself. Instead of seeing life as a series of happy accidents, we can view it as a triumph of information management. The Hamming genome theory suggests that the complexity of an organism isn't just determined by how many genes it has. After all, a tiny water lily can have more DNA than a human, but by how robustly its information is protected. We are essentially walking, breathing databases that utilize advanced mathematical principles discovered by humans only 80 years ago. As we move into an era of synthetic biology and gene editing, understanding these error correction codes is vital. If we try to clean up the genome by removing the non-coding regions, we might inadvertently destroy the very shield that keeps our genetic message intact. The 98% isn't a mistake. It is the silent guard of our biological legacy, proving once again that in the world of biology, nothing is truly junk once you understand the math behind it. For more incredible facts like this, don't forget to subscribe and turn on notifications.